My name is Delara Watan. I'm a first-year graduate student at the University of Colorado Boulder, and today I want to be talking about some of the previous work that I did in the Polymer Lab as an undergraduate and a research assistant. So one of the overarching goals of the Polymer Lab is to be able to image cellular processes using fluorescent microscopy techniques for long periods of time. And as a part of that, my research was to look at host pathogen interactions and develop tools that could be useful in imaging effector proteins using fluorescence microscopy. More specifically, I was interested in looking at the pathogen Listeria monocytogenes and apply some of the tools that have been developed in the lab for other pathogens into the Listeria system. So Listeria is a foodborne pathogen. The infection can be caused by the consumption of contaminated foods. So what makes Listeria particularly interesting is its ability to survive in a very wide range of temperatures and environments. And the pathogen is particularly dangerous because it can cross tight barriers to infect internal organs and even cross the blood-brain barrier and spread into the brain. So Listeria are really good at infecting and spreading the infection, and a reason for this is their ability to, when they infect the cell, it's their ability to really modify the host cell, and they do this by secreting a set of effector proteins that are able to help the pathogen then replicate and spread from cell to cell. And they do this all the while they are escaping the host immunity. So these effector proteins that are secreted into the host cell have a wide range of specialized functions in the cell. Some of them bind native host proteins and inhibit their functions. Others just bind them to block them from interacting with their native protein binding partners. So what we want to do when studying infections is study these effector proteins, and a way to do that is to study their localization so that we can gain more information about their native or about their host binding partners, and also we want to look at the localization changes of these effector proteins during the evolution of the infection in the cell. So normally this could be done, if we want to image any protein, this could be done by attaching a fluorescent probe or a fluorescent tag into the protein, but unfortunately this can't be done when working with effector proteins that are secreted, because the tag fluorescent protein onto the effector usually inhibits its ability to be secreted into the host. So to get over this, our lab has developed a split FP system. So we use fluorescent proteins to tag these effectors, but how we do it is we break the fluorescent tag into two different pieces, so a smaller part and the larger part. We then genetically add the smaller part of the fluorescent tag onto the effector of interest. Then we transiently transfect the host cells with the larger part of the fluorescent tag. So the host is producing the larger part, while the pathogen is only responsible for producing the smaller part as a fusion to the effector protein. So then after the host cells are infected with the pathogen and the effector protein of interest is secreted, these parts, the smaller part and the larger part of the fluorescent tag, are able to then find each other and mature into a fluorescent tag. So this way we're able to image the localization of these effectors over a long period of time, and we also get over the fact that a whole large fluorescent protein attached to the effector would inhibit the secretion of the effector. All right, thank you.